be talking about integrative system framework.
Well, thank you. Well, first of all, thanks the organizer for this wonderful opportunity for us to present some of our work over the last few years. So here I just want to clarify that our sense of a system is really talking in the sense of traditional control theory, signal processing, system modification framework. So in a sense, it's how we really formulate the electrophysiology problem in terms of the traditional signal processing. Okay. So, well, thank you for Nicola give a wonderful keynote earlier this morning, so I don't need to talk about the motivation in terms of the clinical side, why we want to do this. So in a sense, we are more look at, say, from non-invasive observations on the patient in terms of tomographic images and also the body surface potential maps. How can we get more out of it, or how can we make better decisions from those data? So in clinical settings, it really has been very sophisticated and getting more sophisticated pattern recognition process in terms of decipher the information from the available data. And in our intention, say, well, how can better models to help this process and make these information, decipher the information more sensible? Yeah. So our basic premises is say, well, better models probably should lead to more appropriate constraints when you do data analysis. And there's a constraint in terms of the physiological plausibility versus computational, or in terms of algorithm, the feasibility of that. But the other thing, though, is we say, well, can we actually recover the volumetric electrophysiology from body surface potential? Obviously, here we in the Newton Institute, by its Maxwell equation, we know theoretically that's impossible because at the best you can get back from to the epicardium surface. But what we are intending to do is say, well, if we do have the models about the electrophysiological process, then that model can in some way constrain the data such that the solution space is limited, so we can do something over there. Ultimately, as Nicola said earlier this morning, say we have to personalize these models to make the whole process really meaningful. So this is overall our framework. So from the individual subject, we can use the prior models, or prior knowledge about the models, which is obviously in general. And then we use the patient information observed on the patient. In our particular case, are the tomographic MRI images as well as body surface potential maps. And from these, then we try to get back to the individual subject. And so in that sense, we do system modeling in terms of the dynamics and the observation equations as well, and acquire the data. And then obviously the key question is really do the information recovery in terms of the electrical functions, the tissue properties, and hopefully also for the latent substrate. So most of these works are in upcoming TBME paper, which is I think 21, 22 pages long, so I have a lot of details in it. Anyway, so what we look at it is really say, from a system perspective, is really we are using phenomenal or phenomenological models to constrain the data analysis, so in such that the data analysis have a physiologically meaningful constraints. On the other hand, is we use the data to drive the derivation of the patient-specific model parameters. Okay, so these are the two things we're looking at. And some of the background things, since we are actually image processing group, we have done lots of the representation per se. And for the heart, we're using the mesh-free method after the segmentation of the heart from MRI images. We generated mesh, and then in terms of the volume representation, we're actually using point clouds to do represent as a volume. The main reason is really the easiness of defining the fiber directions on the points instead of the finite element mesh. And then we map the Auckland heart model to the ventricular heart model to our structure. And for the surface, for the body surface, we actually use a boundary element. So the main reason is that there are a lot of studies showing that the isotropic and homogeneous volume conductor seems reasonably well, and we actually have done some of the validation of that as well, and it seems the loss or the error introduced by the isotropicness is limited. Okay, so overall, at the end, we have a mixed, three-dimensional point clouds, and for the heart, and they have the boundary element surface for the heart torso model. And from this, we're using, we model the volumetric transmembrane potential dynamics, and then the other side is really say how the TMP are projected onto the body surface. And as I said earlier, we're really looking at this moment. We're more looking at the phenomenal versus the actual physiologically closer, to reality model. And so the main reason is actually it's computationally or in terms of algorithm analysis, it's not yet feasible yet. Okay, so this is a computation based on for the TMP dynamics, and so this is just the time variables for the excitation variables and as well as the recovery variables. And then from the transmembrane potential to the body surface potential, have this mapping, and we're assuming a quasi-static electrical mechanism. So we come up with we can use the Poisson equation. And so basically, from the Poisson equation, we can, either have a direct solution method. Obviously, it's impossible to solve this, or you can have this, mixed point clouds plus the boundary element representation. And from this, this is integral on the surface as well as the volume. And from this, as engineers, we convert it into a state space representation, so-called our system representation. And for the TMP activities, and also for the mapping from the TMP to the body surface. So this is state variables, and this is a measurement equation, and with uncertainties related back to the measurement as well as to the model parameters. And so of all this, we can have a state equation, in very general form, as well as a measurement equation. The problem, obviously, is that it's a very nonlinear, even for this very simplified model. So you need, when you solve it for nonlinear models, there are a lot of issues, particularly for signal filtering or for system identification, and it's minimally in the UV in the dimension of a few thousand. So one way you do it, you actually can do Monte Carlo integration, and you do the prediction as well to do the correction, but this will be computationally just almost impossible if it's really not really impossible. So what we did is really try to find a nonlinear transformation which is reasonably workable. And so what we found is that we're using a combination of the uncentered transform such that instead of you doing the whole thing, you only transfer up to second order statistics. And so it's really a combined of the uncentered, so you use the Monte Carlo integration with deterministic sampling to do the prediction, and then use a common filter to do the correction. Okay, so this is a lot of, so this is how to estimate the transmembrane potential, and this is the equation. Next equation is how to do the parameter estimate in terms of the model parameters. And so we have done a lot of simulation experiment on it, and this is our more recent work on using the PhysioNet. I think this is a challenge data for the challenge for computing cardiology a couple years ago. And so we look at these 4 post-myocardial infarctions data, and so this is what we tested our framework. And the gold standard is use a gadolinium enhanced MRI, identify the infarction area as a comparison for what we can identify. So I will skip all these procedures. So we try to discover the TMP dynamics as well as the tissue excitability, and from that we can have a sense of the electrophysiological abnormality. And so this is simulated a normal TMP for the case 2, a particular geometry of the heart. And so this is recovered TMP dynamics from the MRI structure as well as from the BSP data. And from this, we actually did so. One is if we only look at the TMP dynamics, this black contour sort of indicates where the infarctions are. And then the colored, from red to green, yellowish things, these are the recovered tissue excitability. The interesting thing is that these 2 roughly overlap to each other, but not exactly. So we can see is that there are, the abnormal electrical function or the TMP actually occurs within the infarct zone, but there are obvious discrepancies between the excitability and the TMP dynamics. So we're looking at this, we're further investigating what's the relationship and what's a possible explanation for these things. And also, we did a lot of further analysis of these things, and given times, I'm going to skip this. And these are the things for the other other 3 cases as well, and it's indicated a different thing. And we made a comparison of these things to the gold standard, and the results obviously seems quite good. And we also compared to the existing results on the same data set, and what we can claim is our result is probably as good as the best of the other things for all cases. So in conclusion, we try to from the non-invasive observations of the images and the body surface potential to recover the volumetric cardiac electrophysiological, for both the TMP dynamics as well as the tissue excitability. And hopefully, as we can move on to the latent substrate detection. And for this, we are starting the collaboration with SUNY Upstate Medical Center as well as Rochester General Hospital.